right, everyone. Thank you for being here. Good morning. I'll get started. So my name is Bhaavya, and I'm a graduate student in the Shaw Lab in the Department of Neurobiology and Behavior at Cornell University. And today I'll be talking to you about the ontogeny of mating behavior in Wapala crickets. So based on an organism's life cycle, diverging phenotypes can occur at many different points, from pre-mating and pre-zygotic to post-mating and post-zygotic. In particular, these pre-mating barriers are a lot of interest for speciation studies as they occur early on in the reproductive cycle, and they evolve faster than later barriers and can thus reduce gene flow significantly. Within these pre-mating barriers, sexual isolation in particular encompasses differences in mating rituals, courtship behaviors, sensory traits, and preferences, and it's particularly important. So for example, in these butterflies, females of the species P. occidentalis assess the amount of melanization on the male forewings to distinguish conspecifics versus heterospecifics. At the same time, these traits that are involved in sexual isolation that allow for discrimination between species can also serve as within species signals. So that is, they allow individuals to discriminate within a species, compatible versus incompatible mates. So for example, in these butterflies from the last slide, females of one species use that same cue, the amount of melanization on the forewing, to also discriminate between or choose between conspecific males, as well as discriminating between conspecifics and heterospecifics. However, there's potential link between traits that diverge under sexual selection within a species, also regulating reproductive isolation between species, has not actually been studied in many taxa. So this leads to my broad question of how do intraspecific processes modulate interspecific differences and speciation? And so if the phenotypic traits that underlie sexual isolation between species diverged under sexual selection within a species, then we should be able to identify traits that differ within a species, whether with sex, age, or other factors. And this question is really aiming at a larger problem of how individuals find compatible mates. They have many problems to overcome. They need to find potential mates who are developmentally ready or at the right stage of the opposite sex, and who are genetically compatible, such as, for example, a conspecific. And so my interest is really in looking at how these traits evolve. So an excellent candidate so I'm only going to be talking most of the talk about the behavior, but I just wanted to give you a brief overview of the whole context that my project is in. So an excellent candidate for such a dual purpose trait is chemosensation, which is important in speciation and mate choice across many taxa. But this area of research has primarily focused on insects because they use chemical cues during host choice and courtship, and they are tractable systems for experimental studies. And insect chemical signals can come in a variety of different types, from long range to short range, volatile, non-volatile, so on. But a key modulator of insect chemical communication at close range are cuticular hydrocarbons, or CHCs. So these are long-chain hydrocarbon compounds that are present on the insect's exoskeleton, and their original function is thought to be preventing desiccation, but they are also functioning as behavioral signals in many species, especially for mate choice and courtship. So different species can have highly differentiated CHC profiles, and CHC profiles also change during development in many species, thereby potentially serving as indicators of behavioral maturity. So what would a scenario where CHCs are involved in both sexual isolation between species and sexual selection within a species look like? So say we have this ancestral population with these three peaks. So this is a chromatogram, and on here, the x-axis is time, and that separates out lighter versus heavier compounds, with heavier compounds coming out later. And on the y-axis, we have abundance, which just shows you how much of each compound is present in this overall mixture. So over time, this ancestral population becomes two separate species with two different CHC profiles. Within each species as well, mature and immature individuals have different profiles that differ in, for example, their abundance of specific compounds. So here, CHC profiles can serve both as an interspecies behavioral isolation signal and an intraspecific function that indicates within species mate compatibility, so when individuals are adults and behaviorally ready to reproduce. So an excellent model system to study the relationship between intraspecies processes and interspecies differences is La Pala. This is a genus of sword-tailed crickets spread out, containing 38 species that are spread out throughout the Hawaiian archipelago, and they're the most rapidly speciating invertebrate clade known. They're generally ecologically and morphologically cryptic, as you can see in that image. The most conspicuous difference between species is the species-specific male song, and females have species-specific preferences for that male song that leads to assortative mating. At the same time, there's also a great diversity in CHC profiles across these species, and previous research has shown that these CHCs are used in mate choice decisions. But observations show that there is a period of at least 10 days after crickets molt into their adult forms when they're not actually receptive to mating. So during this time period, males, although they are morphologically mature and they have wings which they use to stridulate to make song, don't sing. Females are skittish and they don't engage with sexually mature males. So lack of male song during this time means that females must rely on other cues to assess compatibility. While since females never sing, males in general need another phenotype to assess females. And courting an incompatible individual could result in loss of time, energy, and gametes, and so selection should favor signals that would allow for distinguishing between mature and immature individuals. And so today I'll be focusing on the changes in mating behavior that marked this transition from morphologically mature to both morphologically and behaviorally mature. And then, again, just a reminder, but this work is in the context of looking at the correlation between these changes in mating behavior and age-related changes in CHCs and investigating if those correlated changes also impact reproductive isolation. So La Pala has a really long courtship ritual, which can last 6 to 8 hours every day. This begins with male song that attracts females, goes on to antennal contact, facing behavior where the crickets face each other, production of a nuptial gift, which we call micro or macro spermatophores, copulation, the female eating the nuptial gift, and then the whole cycle is repeated again up to 11 times during the day. And I want to draw your attention to these behaviors marked with an asterisk because these are behaviors where the crickets are really in close contact and touching each other with the antennae. So antennae in insects have chemosensory organs, and so it seems likely that the crickets are engaging in chemical communication during these behaviors. And so I'm interested in, what does the onset of behavior as individuals become reproductively mature look like in this genus? So I did mating trials using my study species, La Pala macaea, which is found on the island of Maui. I paired individuals of known age from 10 to 12 days post their final molt to 25 to 27 days post final molt with sexually receptive adults of the opposite sex, so there were 6 age groups. I froze the individuals after mating trials so that I could then use them for later CHC extraction. And I recorded 1 hour-long trials starting from the time of first antennal contact. And then I scored behaviors in Boris. So before we go on to the results, I just wanted to show you what some of these behaviors actually look like. So this is male singing. Unfortunately, there's no sound, but you can see that the male lifts his wings up and starts to stridulate them, rub them together to make sound. And this is the female next to him. Then antennal contact is also what it sounds like. The individuals rapidly make antennal contact. They have these really long antennae that can be up to twice their body length, so they can touch the other individual from, a fair bit away. Then we have facing behavior. So they are actually moving, but they just stand close together with antennae occasionally moving a little bit for quite a long time. And this is also really important behavior because this is when the male most often actually makes his nuptial gift, so a microspermatophore. And I also scored this behavior differently if it happened after copulation. So after the male transfers the nuptial gift to the female, they also face, but we refer to that as mate guarding. So I scored those differently. And then finally, here's copulation. So here, hopefully you can see this little white dot, which is the nuptial gift on the male. And the male will be backing up under the female and the female can accept it by basically rising up on her hind legs and allowing the male to fully back up under her. All right, so now that we've seen these behaviors, let me go on to some of my results. So here's singing for the focal males that I had. So in these graphs, the age group will be at the bottom for males or females, and then the y-axis will tell you which behavior I'm scoring. So there was a significant difference in male singing across age groups. 10 to 12 days post final molt adult males actually did not sing. They just lifted their wings up briefly and then put them down. And then male actual singing, so actual stridulation, was observed at 13 to 15 days old. But these songs were generally very brief. So they would sing like less than 5 pulses and then put their wings back down. But after that, males sang fairly consistently, and older males generally spent more time singing. But as you can see, there's also a lot of individual variation. So I trialed 5 individuals for the 16 to 18 day age group, which you can see over here, but only 2 of them actually sang. But when they did, it was very consistently long, regular songs. Then going on to facing, so here we have females on the top panel, males on the bottom. And females generally spent more time facing per each bout than males. And older age groups had more facing than younger age groups. Again, there was lots of individual variation. So like I said before, those 16 to 18-day-old males did not engage in much mating behavior at all, starting with singing as well as facing. But you can see that 16 to 18-day-old females were engaging in facing pretty consistently. Then going on to further behaviors in the ethogram, so looking at microspermatophore production and transfer. So like I said, micros are often produced during or after facing. So the proportion of males that made a micro is generally consistent with the number of males that engaged in facing. So if they were facing, they often did actually end up producing a micro. But in contrast, only 22 to 24-day-old males actually even attempted to transfer the micros to the females. The younger age groups produced micros, and then they just ate it after some time. They made no attempt to actually transfer it to a female or to attempt copulation. We can also look at female micro acceptance. So when the non-focal male, the sexually receptive adult male, who is paired with females of these different age groups. So again, this will be the behavior whether the females raised up on their hind legs to allow the male to back up under them. And if they don't want to, they will retreat or they will just not back up, and so the male has to give up. So in contrast to males only attempting to transfer micros at 22 to 24 days old, females were actually accepting of micros at 16 to 18 days old. This doesn't necessarily mean that the copulation was successful, as you'll see in a moment, but just that they indicated willingness by rising up on their hind legs. All right, and so another way to look at female receptiveness is to look at what the non-focal sexually mature males actually did when they were paired with these females of different age groups. And non-focal males made micros for females as young as 13 to 15 days post their final molt. But they only, again, successfully transferred them with females aged 19 to 21 days or older. So there was this gap between when they would start making micros, and even if they attempted to transfer it to females, the females wouldn't actually accept it until they were 19 to 21 days old. So briefly, my conclusions: both males and females exhibited age-related changes in behaviors involved in courtship. For males, all of these early behaviors, singing, facing, microproduction, start quite early, so 13 to 15 days post their final molt, but transfer attempts and successful matings come last. And for females, facing begins at the same time as males, but doesn't actually indicate that successful copulation will follow. And then we can also look at the mating effort invested by the male paired with the female to look at when a female is behaviorally mature. And again, these initial mating behaviors start at 13 to 15 days post their final molt, but the later behaviors gradually appear over time rather than an abrupt shift, so everything appearing along with the initial behaviors. And the CHC analyses from these age groups and correlation analysis between CHCs and these behavioral changes is in progress. So maybe at next evolution you can hear about that.
So today I'll be talking about acoustic mating displays in Anna's and Costa's hummingbirds. So communication is when a sender emits a signal to a receiver, which enacts a change in the receiver's behavior. The signal has to go through the environment in order to reach the receiver, obviously. So animal communication is used when in context of finding mates, seeking food, establishing territories, detecting prey, and rearing youngs. Sometimes animals communicate in ways that seem counterproductive to their survival. So examples include the long, cumbersome train of the peacock, which gave Darwin a big headache. The long flake plumes of the Goldie's bird of paradise. And so this paradox led Darwin to develop the theory of sexual selection, whereby elaborate traits may aid in reproduction, such as through mate choice. So this sexual selection can result in the evolution of complex signals, which abound in the natural world. So here we have the pygmy swordtail, which sends multiple messages within the visual modality. The bars signal species identity, and the size signals individual identity. In wolf spiders, the signals are multimodal, spanning vibration and visual modalities. When taken apart and played back to the female wolf spiders, they still enact a response, enabling them to be produced in different environmental conditions. However, when played together, they create a larger response. The greater sac-winged bat with a heron produced a perfume that they store in their sacs. They also vocalize and do visual signals. And the satin bowerbirds have extended phenotypes in which they produce bowers and display within them, complexly interacting with males. So I'll be talking about hummingbirds, which have a variety of complex signals, from this spectacularly colored, vibrant violet ear hummingbird to the ruby throat, which flies back and forth in front of other females, to the wedge-tailed saberwing, which individuals can sing over 45 song types. So I'll be talking about the bee hummingbirds, which both sing and dive to females. So here are two phylogenies of the bee clade. Black is the presence of a trait, and white is the absence. So we see that most hummingbirds in the bee clade dive to females, with likely diving being at the base of the tree, whereas singing is less common. A majority of the birds in the clade do not sing. So we'll be talking about Calypti costae and Calypti anna, or annas and costas, which are boxed here. So they both sing and dive, and there's evidence that both species learn their songs. So Costa's hummingbirds do a shuttle display in which they, like this male that looks like an octopus head, flies back and forth, undulating his wings while he sings. They also perch sing and do these dive displays in which they fly up and then dive, spreading one side of their wing at a time. And because they have a specially dimorphic tail, which is this R5 here, it's slightly notched, and that's when it reaches a particular velocity, it creates sound. Its sister species, Anna's, also sings a very high-intensity, close-range song, and they also do a dive. The dive in form is slightly different. They fly up and do a U-shaped dive, and rather than spreading one side of their tail feathers, they spread the whole tail feather. So there's an apparent match in signal form between the song and the dive sounds that they create in both species. So here is Costa's. Here, the spectrogram of their song. So spectrogram is sort of analogous to reading music. On the x-axis is time, on the y-axis it's frequency or pitch. So you see that over time it's just this like upsweep of frequency and downsweep in both the song and dive. This is what they sound like. I'm not sure what happened since then. But if you'll just have to take my word that they sound very similar, and they're very high in frequency. Anna's hummingbirds also have songs and dive sounds that resemble one another. They're slightly more complex, involving multiple components. Also won't play. So males, this leads to our question about why there is a correspondence between the two signals. So are males matching their songs to their own dives? That is, I set out to see whether dives and songs are co-varying with one another. So to do this, I went out and elicited songs and dive displays from hummingbirds in 2 different locations. Anna's were recorded in Park, Sycamore Canyon, and Riverside, and Costa's, a desert-dwelling species, was recorded in the Colorado desert. So I elicited dives and songs from each individual and then scored in Raven Pro components of the songs. We then took the individual mean for variable measures and tested within individual correlation between song variable and corresponding dive sound. And I couldn't have done this without the help of 2 really great undergrads, Wendy and Ashley. So the measurements I took were similarity measurements. They're boxed here to show the ones that I was comparing. So pulse rate in the song and dive, this little gap in time where there's no sound in both signals, these 2 whistles, and then the dive sound itself, and a last whistle for Anna's. For Costa's, I measured the upsweep itself, the apex of the song and dive, and the downsweep. I also measured pulse rate, in which if you look really carefully, the songs also match in the frequency modulation that they have. So for Anna's, I found that 3 variables co-varied between dive sound and song. On the left is pulse rate. In the middle is dive sound peak frequency lower whistle, which is the green box. And the farthest right is dive sound delta frequency lower whistle. So there's 3 positive trends where it seems like males who produce a faster pulse rate also sing a faster pulse rate song. The same is not true with Costas. So 0 of the variables that we measured co-varied between the dive sound and song. The most closely to co-varying is the dive sound pulse rate. And here are 2 examples of other variables that did not co-vary. So my results for Anna's 3 traits co-varied, and in Costas none did. So conclusions to date. Our data suggests that 3 components of Anna's song and dive sound are co-varying with individual, whereas all the others are not. It suggests that no components of Costa's song and dive sound co-vary with an individual. So what's going on? And I'm hoping some of you can help me try and figure that out. So future directions are to look at how individual songs compare to a neighbor's dive sounds, particularly when juveniles are learning their songs. So I said that Anna's and Costas are both song learners. What I didn't say is that they have a promiscuous breeding system, and so males do not help with parental care at all. So who the juveniles are learning from and when they learn it is unclear. Similarly with dive sounds and dive displays, the pattern of which they learn it is unknown. And so, does variation in dive sounds and songs have geographic trends? And do females prefer males who match their song to their dive sound? It would be nice to also replicate this in other species who have similar patterns of correspondence within mechanistically distinct signals, to see if we see covariation in that sense. So I can allude to what I think is going on. which is there's differences in complexity between the two songs, right? So, and also the dive sound. So in the left is Anna's, which have actually a 3-phrased song in which the terminal phrase matches the dive sound, whereas in Costa's, the whole song matches it. We did find geographic variation in Anna's song, but what we found was it only occurred and was driven by the phrases A and B, but not the dive-like sound phrase, indicating that there is, in fact, some sort of selection to maintain the match. In Costa's, we found no geographic variation, and if you see on the map, all the spectrograms look very similar, right? So there's no variation in song form. So potential reasons for covariation or reasons for not covariation are mate choice. Maybe the females don't really care about matching. Gotta ask the females, right? Ontogeny of learning, where males are learning how to dive or who they're learning how to dive from. Do they even learn how to dive? Who they're learning how to sing from? When do they learn? Lots of questions. And to compare with other species, it turns out that these apparent matches are not only in the Calypdi species, but they occur in a couple other hummingbird species that are not closely related. So the Peruvian sheartail has a dive tail sound that resembles the song, and the bumblebee hummingbird also has a wing trill that resembles the song trill. So with that, I'd like to thank all of my collaborators and mentors.
Awesome. All right, I'll get started here. So my name is Gabrielle. I'm a PhD student at the University of Denver. Thank you for coming to my talk this morning on how permissive female preferences may be facilitating novel signal evolution in the Pacific field cricket. So we see many diverse signals in nature, just a few of which I've pictured up here. And these signals are often used in communication and contribute to the origin and maintenance of biodiversity. And signalers and receivers exhibit tightly knit relationships that can impact the evolution of these novel signals. So signals must not only be detectable to a receiver, but they also must elicit some sort of response from those receivers. So as an example, say we have these jumping spiders here, and the males traditionally have these blue stripes, so the female on that y-axis exerts a high level of preference and acceptance for those blue striped males. If we have a novel red stripe emerge, the female might not accept that new red stripe, and that would lead to the red stripe being eliminated from that population over evolutionary time due to those pre-existing female preferences. And so this tight coupling of signal and preference makes it hard to imagine how novel signals actually emerge and then stick around in populations. And one commonly overlooked mechanism that doesn't really rely on this tight coupling of signal and preference involves permissive female preferences. So female preferences are often very variable, and a permissive female would be accepting of a broad swath of novel songs and signals, and then this would make them therefore more accepting of any novel ones that might emerge. And so this could potentially facilitate the evolution of novel signals by allowing some of these to stick around long enough for more directional traditional preferences to then evolve in a population. So under what circumstances might these relaxed preferences emerge? So selection could be one mechanism that's favoring the relaxation of these preferences, and plasticity could also be contributing to this. And I'm going to break these two mechanisms down in just a few slides with my study system as an example. So to unpack some of these mechanisms, I capitalized upon my study system, the Pacific field cricket. So these crickets in Hawaii are evolving all these novel songs in response to a deadly eavesdropping parasitoid fly. So this fly uses the male's song to find those males and then eventually kill those hosts. And I'm showing on the right-hand side here, we have the ancestral song, but then 5 novel songs that have evolved in just the past 20 years. I'm showing the wing morphs here. So we're seeing rapid evolution in novel signals in these Hawaiian populations of this cricket. So here is the ancestral wing. That ancestral wing produces a loud tonal song using those wing structures that are highlighted in blue and green there. But about 20 years ago, we started to see the emergence of these silent wings. So they actually lost all of those sound-producing structures and don't produce any audible sound. Then, more recently, we've seen the emergence of even more novel signals. So purring and rattling have been discovered in the past 7 years. I'm showing those wing structures there that are modified, and then on the bottom, those spectrograms that are showing that these songs made by purring and rattling males are quieter, more broadband, and differ in a lot of signal characteristics from that ancestral song. So, to investigate how female preferences might be facilitating these novel signals, I capitalized upon the geography of this system. So, these novel songs are in Hawaii, but the crickets are actually from Australia originally. They were brought by Polynesian settlers across the Pacific, so colonized island after island, consecutively bottlenecked, until they made it all the way to Hawaii, where they encountered that North American parasitoid fly that I mentioned earlier that kills any males singing those ancestral songs. So looking at selection, and one case in which selection could maybe favor relaxation of preferences in the system is related to that island colonization process. So Kanashiro's effect, he came up with this effect and this idea that in small, recently founded island populations, females with relaxed preferences might actually be favored because if you have really choosy females that make it on islands, they might actually not be able to find acceptable mates among the founders that made it there with them. So over consecutive bottlenecks, this might select for more relaxed preferences in females. Additionally, I mentioned in Hawaii 20 years ago, in some populations, silence emerged. So some of these females and males have been evolving in completely silent populations, and this might have exerted extra selection just in Hawaii for females to just be more accepting, given the male types that are available there. Additionally, plasticity could also contribute to this relaxation. These crickets inhabit a broad range across the Pacific. Lots of environmental variables differ across that range. Just some examples: acoustic environment, mate availability, and these factors have already been shown in studies to induce plasticity for females when they're raised in different acoustic and mate availability environments. So there is suggestive evidence that crickets in Hawaii may have relaxed preferences, but in the context of courtship. So if you saw Bhaavya's first talk, that courtship, those amazing courtship videos that she showed, this is those close one-on-one context mate choice environments. And it has been shown that Hawaiian females are pretty accepting of all the novel song and morph types in those close one-on-one contexts. But these crickets are out in big fields at nighttime, they're nocturnal, and they actually have to find each other before they have those courtship interactions. So I was really curious about investigating the calling song, what the males use to attract females from afar, and how female preferences for that might be evolving. And so to look at what role selection and plasticity might be playing in these permissive female preferences, I combined both lab and field experiments. So I conducted field experiments in 6 locations, 2 in Australia, 2 in French Polynesia, and 2 in Hawaii. And then to get at that plasticity component, I then conducted this. I brought the eggs back to the lab, raised them to F2 generations, and then raised them in common garden and ran the same test to see how much of my results might be due to some form of plasticity there. And the data I was collecting were phonotaxis data. So this is where I played purring or rattling, those novel songs, silence, or the ancestral song to the females in a little arena, and then just noted whether they went towards the song or not. And to illustrate why this geographic approach is so important to distinguish between these hypotheses, I'm just going to go over some hypothetical results of what would show each of these different mechanisms before showing my own results. So if Kanashiro, that bottlenecking effect, was playing a role here, we would expect high levels of responsiveness to that ancestral song across all the populations. But then we might expect low levels of responsiveness to the new song in Australia, and then intermediate in French Polynesia, been through some bottlenecks, and then the highest levels in Hawaii, which have been bottlenecked the most and had the most selection from that Kanashiro effect. I also mentioned subsequent selection from the fly could be at play here. So if we see even higher levels of responsiveness to these novel songs in Hawaii, maybe the fly is exerting even stronger selection there in those populations. And plasticity, I could kind of go in a bunch of different ways. So I'm basically looking at just differences between lab and field data would indicate some role of plasticity in impacting relaxation of preferences. So I'm going to show my field data first. So on the x-axis, I have those 6 sites that I mentioned. On the y-axis, proportion phonotactic, so that's females going towards the novel signals. And I do find a significant effect between population and song. So I'm going to break down, walk through each song to kind of tease apart what's driving those effects. So looking at silence first, we see no significant difference among the populations in response to silence, which is what we'd expect. That's our negative control. I have purring in purple here, light purple here. There were no differences in responses to purring. We do think it's a very, very quiet, attenuated song. We often just see low levels of responsiveness, even in Hawaii, to that. So we think that's, just due to how quiet that novel song is. If we look at rattling, though, in the deeper purple, we do see that Hawaiian females exerted high levels of phonotaxis. There were intermediate levels in French Polynesia, and then super low levels in Australia to that rattling. And then if we look at ancestral, we actually see that same pattern. So super high levels of responsiveness in Hawaii, intermediate French Polynesia, and lowest levels in Australia. So we do see some relaxation of preferences kind of following that geographic pattern here in our field data. Now, I mentioned that I ran these in the lab to tease apart how much of that was plastic. So looking at our lab results, we actually see that significant interaction between population and song completely disappear. So there's no significant difference in responsiveness to these novel song types in the lab environment. And these crickets were all raised in silence in common garden, so they didn't hear any songs while they were developing and everything before I tested them. So definitely some plasticity at play here, and I wanted to tease it apart a little more, look at what might be driving this loss of differences in preferences among the populations. So I looked at the difference between lab and field data within each song type and within each location. So I'm going to flash a heat map up here, but just to orient you, darker colors are going to indicate the crickets were more phonotactic in the lab. I've highlighted like that 0 point, which would indicate no plasticity here. So anything that's darker indicates that higher difference between the lab and field data. And so when we look at this, we do see some of that darker color aggregating in the top left-hand side. So definitely differences between lab and field indicate plasticity, and we're seeing that a little more, primarily in those ancestral and rattling songs, especially in Australia and French Polynesian populations. And why might this be happening? One theory that I have is there are differences in acoustic environments between these locations. So in Australia, there's no fly. All the males are singing ancestral songs, so the females are developing in these fields with really loud males singing ancestral songs, whereas in Hawaii, a lot of our populations don't have ancestral song anymore. They have all these novel songs, so they're just developing in an environment that's very different acoustically and doesn't have as much ancestral song around and everything. So that could be impacting the plasticity that's happening in my field data. All right, so overall conclusions. Overall, females do demonstrate lower levels of responsiveness to novel songs, especially rattling in Australia, intermediate in French Polynesia, highest responses in Hawaii. And the lab versus field data do suggest that plasticity might be contributing to this pattern. So relaxed female preferences that are facilitating these novel signals evolving are likely both using genetic and plastic mechanisms here. I would like to thank my lab group, my funding sources, and I might have time for a question or two if anyone has any.
I was just wondering, are all of these new songs coming from new mutations, or do you see some level of standing variation in song that these can actually evolve from? Because it seems like really quick evolution for totally new mutations to come in.
Yeah, it is a great question. I do have genomes that, that's going to be my last chapter. I have to learn how to analyze that all. We're all super interested in this. I do think it's from likely different mutations, just from breeding experiments and everything. But that is a great question. And
Variation in the Australian population at all?
Nope, literally none. Yeah, yeah. And the French Polynesia too. They're all categorically ancestral if you analyze the wings using the same methods. Yeah. Yeah.
This is a super cool talk, by the way.
Thanks. Oh, thank you.
I just wanted to ask about why you chose to do essentially a no-choice phonotaxis test, by just playing them one song, whereas typically in other literature you see a binary choice or even sometimes multiple choices about why you're choosing one rather than measuring their preference for one or the other. Because I imagine that in Hawaii there's multiple call types that are coexisting in the same area.
Yeah. I think that is a great question. I think, especially when purring eval, we were really curious about preference functions, kind of manipulating different variables to see what they get. I think it would be really cool, though, to do them in choice trials as well. Have you done choice trials, Jay? Yeah, Jay has done choice trials for sure. So yeah, you can talk to Jay too if you wanted to get any results on that. But that is a great point. I think, I always struggle a little too with acoustic environments and not having songs overlap too much too as well. But yeah, if you have experience in that, I'd love to chat. Yeah. I think. Okay. Yeah, oh yeah.
The ancestral crickets never experienced the novel morphs, but have you ever considered playing a completely novel morph that even the Hawaiian population has never heard before? And what do you expect would happen there?
That's a great idea. I think as long as we have done this before, I think as long as it's in the pattern of the song, Hawaiian females tend to regularly accept it. I haven't tried that in Australia, though, playing a xylophone or things that we played before in that pattern. That I have not done. But yes, in Hawaii, they do tend to accept anything that's played in that same pattern. Yeah. Awesome. I think we're start. I know there are no choices.
All right, so I'll go ahead and get started. My name is Callen Inman. I'm a graduate student with Molly Cummings at UT Austin. And today I'll be talking about how ecology and mating system might shape behavioral and cognitive traits in two syntopic pairs of pieciliid fish species. So broadly, this work falls into the category of interactions between natural and sexual selection. We know of many examples across taxa, including migration patterns and mate choice shaping the degree of sexual dichromatism in orioles, predation and male harassment interacting to affect sexual segregation in guppies, and thermoregulation necessity causing female sea lions to clump at high densities, changing male mating tactics. And I primarily think about these interactions just because of the work I've done from the perspective of sexual selection. So obviously there are many different continua we can think about in terms of mating tactics that males might employ. But one very important one and relevant to the work I'm going to present is from cooperation to conflict. So we have mating systems where males display to females. Females can exercise active precopulatory choice on the one end of the spectrum. And on the other end of the spectrum, we have systems where males actively try to push females above their optimal mating rate, resulting in these forced copulations in the form of coercive mating. And so I'd like to ask with this work, what are the consequences of variation in male mating tactics for the evolution of female behavioral and cognitive traits across contexts? How do females respond to variation in these mating tactics across species? And a great system for looking at this is pieciliid fishes. So they have very high variation in male mating behavior. Many species lack display or cooperative mating entirely, and males force females into mating. A smaller fraction of species employ both coercion and courtship. So this allows us to both look at mating tactics and how females respond to them from the cooperation to conflict dimension, as well as the number of mating tactics that males are employing. And usually when they employ multiple mating tactics, males come in these discrete morphs, and so there are different morphological traits that are associated with these behaviors. So the first study that I did was with 4 species sort of evenly distributed across the clades, so 2 species that employ only coercion and 2 species that employ a mix of courting and coercive tactics. These species had been housed in these outdoor mesocosms that are predator-free but experience other ecological conditions such as invertebrate communities, anurine tadpoles, weather, etc. So predation is one of the main factors that differentiates these populations from wild populations. In these 4 species, we found significant movement differences across species. Females from coercive taxa tended to move less than females from these complex mixed tactic species. And also complex taxa, the mixed tactic species, tended to perform better in inhibitory control from cognitive flexibility, which was consistent with our predictions about how social complexity might affect that cognitive trait. So the study that I'm going to present today is a follow-up study on this, where we look to see whether we can extrapolate from these predator-free populations to wild populations. And so what we did is we selected species that are syntopic, so they're in the same shoals, they have similar foraging ecology, they're experiencing the same predation pressure, but differ in male mating tactics. We assessed syntopic species pairs from 2 locations. One syntopic pair was in Ecuador and one in Texas, and we focused on these traits that we found to vary across mating system in the predator-free environments. So we looked at movement behavior and inhibitory control in females. And so these are the 4 species that I looked at. 2 of them are from this high predation location with a high diversity of piscivorous fish, and one are from a moderate predation location where, due to overfishing, there's a relatively low diversity and abundance of piscivorous fish species. For methods, we used a detour reaching task to measure inhibitory control, the facet of cognitive flexibility. So females have to inhibit this prepotent response to move directly towards this transparent barrier and have to learn to circumnavigate it to reach a female reward. And then we use the Scototaxis tank, which essentially is a simulated emergence from shelter assay, where the white side is aversive and simulates an exposed area, and the black side tends to attract the fish more and is simulates a sheltered area. So we looked at stress movement, immobility, and anxiety, or the time they spend in the sheltered area. So for stress movement, so to reiterate, in the predator-free environments, we found that all coercive taxa tended to move less than the complex taxa. When we add predation to the equation and look at these 2 syntopic pairs, we find no differences between members of the same syntopic pair and no differences between locations in this trait. In inhibitory control, so in the predator-free environments, we found that complex taxa tended to perform better on this task than the all-coercive taxa. When we add predation, we find no differences within syntopic pairs or between locations in this trait. Lastly, we looked at this trait anxiety, which we had not focused on in the previous study. But interestingly, in this trait, we found contrasting patterns between mating systems by location. So at the high predation location on the left, we found that the all-coercive species showed higher anxiety, whereas at the moderate predation location on the right, the mixed tactic species showed higher anxiety. So is this a mating system by location interaction? So we looked a little bit more deeply into this, and we had to take into account body size. So unfortunately, for making inferences about mating system, there are body size differences across species, and the all-coercive taxa tend to have smaller body sizes than the mixed tactic species. Interestingly, and I have this circled. When you look at body size and anxiety correlations, and this is showing body size and anxiety in the high predation location below and the moderate predation location above. You see this interesting pattern where all coercive species tend to cluster when they have the same body length at a similar anxiety level, whereas the mixed tactic species tend to differ. So why might this be? So to get greater confirmation of this pattern and to try to differentiate whether we're seeing a mating system by location interaction or a body size by location interaction, We constructed multiple GLMs with different combinations of mating system, location, body size, and different interactions. And the two best fit models were one just containing mating system by location and one containing body size by location interaction. And there's maybe slightly more support for a mating system by location interaction based on the AIC weight, but really we can't differentiate between these two variables based on these GLM results. So we can explore these two possibilities. So what is it? So if there's a mating system by location interaction, what does this mean, and what do we how does this differ from what we might expect? So unfortunately, prior work in Piscileids is inconsistent on this issue. So for example, in populations of female guppies that have higher rates of male harassment, they show greater emergence from shelter. But the opposite is true in experimental populations of the El Abra swordtail, and work on other species such as Brachyraphis is also inconsistent on this issue. So unfortunately, we don't have very refined predictions on how mating system might shape anxiety and how that interacts with predation, so we really need replicate populations to get at this question. For a body size by location interaction, we have a little bit more support from prior literature in terms of people advancing hypotheses and those being supported. So in Brown and Braithwaite 2004, they summarized two major hypotheses about anxiety and body size relationships in small fish. So the first is a predation hypothesis. So this predicts a negative correlation between anxiety and body size. Smaller fish tend to be more vulnerable to piscivorous fish than larger fish. So they should spend more time in shelter. On the other hand, a metabolic hypothesis, taking into consideration the faster metabolic rate per gram of smaller fish, lower body fat reserves, they should have greater foraging motivation, and so smaller fish should be faster to emerge from shelter than larger fish in the absence of predation. And so what we find in our results is that the moderate predation location pattern is consistent with this metabolic hypothesis, that smaller fish tend to be less anxious. And on the other hand, the high predation pattern is consistent with the predation hypothesis, that smaller fish should be faster on average to emerge from shelter than larger fish. And so this is at least suggestive of this pattern by predation, where predation is sort of swamping this expected metabolic relationship, but we need further replicate populations to confirm that this is true. So overall, when we look at wild populations, female Piscileid fish are not showing the consistent differences by mating system in behavioral and cognitive traits that we saw in the semi-natural populations. Instead, we find possible interactions between mating system and ecology or between body size and ecology. We really need new data to confirm whether this is true in shaping the anxiety response. And so current work is exploring this more comprehensively. So I'm looking at the same syntopic pairs, but multiple populations of each one, looking at both sexes to differentiate between female-specific and male-specific responses, and looking at them under the conditions of multiple predation regimes in different watersheds to really try to tease apart these questions. So with that, I'd like to thank my lab, the Cummings Lab, various funding sources, undergrad assistants, and my dissertation committee.